In the final session, my main feeling is everything has been said today, but maybe, just maybe, I can add some of the stuff that we struggled with the last couple of years using EVs as biomarkers. And today I'm just going to use bladder cancer as an example of what do you have to adjust, what do you have to take into account when you are working with clinical samples, and a lot of them, and you want to analyze, you want to isolate and characterize the EVs and do biomarker research on them. So, very short, bladder cancer, why are we working on this? We always start from basically a clinical need. We get into contact with clinicians that have problems diagnosing their patients. And the urologist that contacted me, he was kind of worried because most of the techniques that are used today or lack sensitivity or are very expensive and invasive, and they were looking for alternative methods to use to screen patients for bladder cancer. The reason why we thought it was a good idea to use extracellular vesicles as sources of biomarkers was because we know from literature that EVs are secreted more by tumor cells than by the healthy cells. So if we enrich the urine for EVs, that we do basically a relative enrichment of tumor material. So we thought it was a good idea to focus on EVs and more specifically on the protein content of these EVs. When designing an experiment like this, there is a lot you have to take into account to, and it's basically the whole session of today you have to take into account. What is the optimal collection of the urine samples? What methods do you use to isolate your EVs? Your experimental design, how many samples do I need to include to get statistically relevant results, and how do I normalize my data? When do I know that the quality of what I produce is actually well? So I'm going to take you through this whole process of basically experimental design, experimental optimization, and I want to address myself to all the PhD students and the graduate students that are in the audience. Basically in our lab, the PhD students, they spend half of or more of the time of their PhDs optimizing protocols. And basically, it's in the last year of their PhD that they have to run all the samples and do the data analysis and get to the results. But I think that's normal, so hang in there. It's good that you focus on the optimization, because if you start your final experiment and you start running samples, there's no way back, basically. You have to analyze all your samples the same way, do it in a reproducible way, and once you kick off, there's no way back. So, as a lot of people already told you today, I'm going to reference to the ISEV guidelines, basically, especially in this case, the urine extracellular vesicle guidelines that have been published by ISEV. Also, our own results that were also published in JEF. First, your patient information. What do you need to know about the patients you include in your cohort? Well, basically, this is the minimum, basically. You need to know the age, the sex, where did you collect your samples. But of course, if you have outliers at the end of your experiment, you also want to go back to clinical details. So that's not included in this table, but you also want to know the staging of the tumor, the therapeutics they already received, did they smoke, all of these things. Also the facts about your sample. How long was your sample basically on the bench before it was frozen? All of these things, register it so that at the end of your experiment, if you find some odd results, go back to all of those data and find out where it went wrong or what happened in your analysis. For the urine collection, when you work with real clinical samples coming from the clinic, we know the list of these best practices that have been published in these guidelines, but what we ran into was that if you work with university hospitals, it's easy to get banked samples or to get them centrifuged and stored at -80 directly. But if you go to smaller hospitals, which sometimes also have a very large cohort of samples available, you run into trouble because they don't have a centrifuge in the lab to centrifuge the samples before to storing them. They don't have a -80. So basically you have, in some way, you have to not compromise. You have to do it in a good way, but do it the same way every time. Make a protocol that is reproducible, and indeed basically put in your results what you did, describe what you did. I think that's the most important thing. For us, we used single void urine, basically random urine, not morning urine, but basically because a biomarker in the end, you cannot tell your patients, yeah, we cannot run the test because it's evening. Yeah, basically a biomarker has to work all day. So also that take into account when you design an assay, that you have to be able to use it, and that it is not ideal from a scientific kind of view, but it is not usable in the clinic. So there you have to take that into account when you design your experiments. So we use single void, no morning urine. We checked the urine quality, we checked for hematuria. We stored the samples before centrifuging them at -20, which is not ideal, but was the only way to collect the number of samples we needed. We thawed and centrifuged them afterwards, and then aliquoted them and put them in -80 for further use. The isolation, there was a question today from someone, well, if you need to analyze thousands of samples, which method is usable in a clinical setting? At the time that we started off this project, the SmartSec kit was not available, but today there is like a 96 well format of size exclusion chromatography that you can use for large amount of clinical samples. It works quite well. We use it for CSF. In this case, we had large volumes of urine, so we chose to filtrate them first. Filtrate them to around 500 microliters and put that on a size exclusion column to do the final isolation of the vesicles. The part about the characterization, I'm not going into detail. We heard about it today a lot, NTA, Western blotting, microscopy. Maybe one interesting technique to mention, because I don't think it has been mentioned today, is AF4, the asymmetrical flow field flow fractionation. It's a nice way to combine it with the NTA because it has a UV detector. And if the line basically is flat, then you know that the protein content in your sample is below the limit of detection. So that probably your protein contamination in your sample is quite okay. And then in one run, you can also see what is the size of your vesicles and basically the concentration of the fraction. So it's a nice and complementary way of characterizing your sec fractions. Then the experimental design, looking at from a proteomics point of view, it's also been published. I think a very important thing in biomarker discovery, especially using proteomics, is your variation analysis. What we did before starting our big experiment was actually measuring what is our instrumental variation, what is the technical variation, the intrabiological variation, and the total intrabiological variation. The setup of the experiments is displayed here, so the result is more important. If you see, for instance, here, the line of the intrabiological and the total intrabiological variation, it's almost equally high. So that means that if you take urine samples from one individual, but from different time points during the day, the variation of your proteome is almost as large as you take urine samples from another individual. And that's quite important to realize, because that means you will have to include quite a lot of samples in your cohort to make sure that you have statistically relevant results. So this table is just a generic table basically for the parameters that are described here. Depending on the effect size, so the fold change of your protein, if you take a look at the lower row basically of the table, you see that depending on the effect size, when it gets smaller, you need to include quite a large number of samples. Other ways to limit false positive results is what we did, because it's very difficult to get 400 samples from a clinic to do your first experiments. Is you can also do a manual check on your raw data from your proteomics experiments, check the peak shape, check the quality of your fragmentation spectra, filter out results that you think from a quality point of view are not sufficient, and also exclude proteins that are based just on one unique peptide. Just some results. This table is what I already showed. In the first biomarker study, we wanted to find a biomarker that was able to diagnose a first diagnosis, so people that were healthy in the past and that have a first tumor. How do you diagnose them? And in the second setup, we compared patients that basically relapsed from cancer, because in the case of bladder cancer, these chances are very high actually. And we compared them with tumor-free samples after diagnosis, so to be able to find a biomarker to check for relapse of bladder cancer. The workflow, basically in total, we analyzed almost 300 samples. We filtrated them, isolated the vesicles, we characterized all the vesicles. We did a protein extraction, and as already mentioned also by colleagues, Suresh, who is also an expert in proteomics, you can do it in several ways, and depending on the protocol you choose, probably your output will also be different. You can use solvent extractions, you can use S-traps. Basically, most of the methods that are described are fine, but you have to make sure that for the sample type you use, you optimize your method and you make sure that your output is optimal. We analyzed some on mass spec, and the results were that we identified and quantified over 1,200 proteins, and then in the first study setup, we identified almost 200 proteins that were differentially expressed. In the second setup, that was only 4 proteins, of which 2 were hemoglobins, and which are not really the most interesting biomarkers, because probably they are the result of the blood that was still present in the samples. Of the almost 200 proteins we identified, we also did the same quality control of the raw proteomics data, and we kept almost 70 proteins of wrong way. And we did a pathway analysis and a string analysis, and basically we found that a lot of the processes, or some processes that are really linked to EVs, are apparently associated to bladder cancer. But this, I'm not going into detail into the biology of the results, because there are some small points to wrap up that I want to bring to you from a proteomics point of view. So if you have large cohorts of samples, what is a good way to analyze them? You have to block randomize your samples. It means throw all the diagnosis and the centers you collected them from, mix them in a way that there is no way that you analyze, for instance, all the samples coming from hospital 1 in one batch and the other ones later, because that's a way to get extra confounders in your analysis. Also, use reference samples. It's quite important that if you analyze 300 samples on a mass spec, that you know from the beginning of your experiment and the end of your experiment that your reference sample is still the same. So at the right side here, you can see a typical batch scheme that we use to analyze samples. So you see that there are blanks included, pool samples included, also HeLa digests included. It's kind of a reference, an easy reference sample, which you know what you have to identify. Also check below if the peptides or the proteins you identified, if the retention time of your peptide stays the same, because if you want to quantify them and your retention time shifts, it becomes very difficult for the software to know where the peptides are eluting from your column. And also if you see, for instance, that the number of master proteins over all of your batches, you see here from left to right, going from batch 1 to batch 16, you see a little bit of decline in the number of master proteins identified. If you know that, you can go look for the problem. And in our case, the problem was that we had a little bit of polymer contamination. But then if you know that, you can try to fix it. If you don't know it and you basically close your eyes, then you don't know in the end what you're looking at. So the take-home message is basically, yeah, we have another poster, but I'm going to skip that. The take-home message is spend enough time on optimizing your experimental design. Take the time to optimize everything and report everything that you did. Thanks.